In episode 1, we learned of a few ways humans could have become infected with infectious diseases like SARS and MERS coronavirus. But let's refresh those ways for those who may have missed episode 1. Now, SARS 2003 evolutionary origin supported a recombination event of parent viruses between avian and mammals, meaning an avian virus had mixed with a bat virus that then evolved into a new strain of virus like SARS, which was capable of infecting humans. Now, how the virus may have spread to humans could be from humans interacting with bats in their environment, like visiting and touring bat caves and then coming into contact with contaminated bat guano, or through zoonotic reverse zoonotic cycles, where humans are capable of infecting mammals with their viruses that then evolve into newer, deadlier strains of the original virus and then is reintroduced back into the human population. Now, even though those are actual natural events that do occur and can cause emerging infectious diseases like SARS or MERS, they do not eliminate the possibility of laboratory involvement. As the same way these events can happen naturally is the same way they can happen through experimenting and creating chimeric viruses in labs. Which leads us into, could COVID-19 have been created in a lab and then accidentally or intentionally released? A chimera virus is a microorganism created by joining nucleic acids from two or more different microorganisms. Combining two pathogenic viruses increases the lethality of the new virus, which is why there have been cases where chimeric viruses have been considered for use as a bioweapon. For example, the Soviet Union's Chimera project attempted to combine DNA from Venezuelan equine encephalitis, smallpox, and Ebola virus in the late 1980s. Biological warfare is the use of toxins of biological origin or microorganisms as weapons of war. For example, anthrax, glanders, the bubonic plague, smallpox, etc. Now, recently, there has been widespread speculation on whether COVID-19 could be a chimera bioweapon. Now, the assumption is that given the novelty of SARS-CoV-2, it's unlikely to be a bioweapon because recent research suggests that the virus is likely natural in origin, although the immediate natural reservoir of the virus is yet to be identified, allegedly. Additionally, it would seem improbable that a bioterrorist would use an unknown natural virus as a weapon unless they were involved in experiments that ascertained that such a virus or one of its ancestors could effectively bond to human receptors and infect human cells. Well, isn't that interesting? Because that's exactly what happened in 2008. After the SARS outbreak of 2003, around 2004 or 2005, Chinese horseshoe bats were discovered carrying SARS-like coronaviruses. So before 2008, a study was conducted to determine if these bats were the natural reservoir host of human SARS coronavirus. So they used an HIV pseudovirus-based system with cells expressing ACE2 to test the S protein of the bat SARS-like coronavirus. Now, since SARS human coronavirus uses ACE2 as the receptor binding to gain entry into cells to cause infection, this is why they used HIV cells expressing ACE2 to test the S protein of the bat SARS-like coronavirus, because the S protein is what's used to help with the receptor binding. If the bat SARS-CoV S protein cannot bind to ACE2, then it would mean that these bats were not the direct progenitor of human SARS-CoV, even though they carry a SARS-like virus. These bats they found were not the direct progenitors, as they could not bind to human or civet ACE2, but it did point to bats as being the possible natural reservoir host of SARS that caused the spillover event to humans, and that SARS may not need an intermediate host like the civet to cause infections to humans. Now, here's where things get interesting. After they injected the HIV ACE2 cells into the backbone of the S protein of the bat SARS-CoV to determine its receptor binding capabilities and figured out that it couldn't, they decided to inject a minimal amino acid into the S protein of the bat SARS-CoV, which enabled the bat SARS-like coronavirus to now be able to gain entry using ACE2, which means they effectively created a chimeric virus capable of infecting humans. Now, if you think that's damaging, that's nothing compared to the article that came out claiming that COVID-19 had HIV proteins in it. And not only that, they found 4 new inserts in the S protein of COVID-19, which was quite surprising because it wasn't found in the S protein of SARS 2003. And it is quite unlikely for a virus to have acquired such unique insertions naturally in a short duration of time. And even though this article was withdrawn with claims that this wasn't true, it's a little too coincidental that the same experiment they did in 2008, where they injected amino acids into the S protein of the bat SARS-CoV, and they used an HIV pseudovirus-based system with cells expressing ACE2, makes one wonder, is this claim really false? I'll let you guys speculate on that. After these bats were determined not to be the direct progenitor of human SARS-CoV, the search continued on for more bats that could possibly carry the SARS-like coronavirus responsible for SARS. And in 2013, they did find 2 new bat SARS-like coronaviruses that were more closely related to human SARS than any other bat SARS-like CoV they had found so far. Bat SARS-CoV RS3367 and Bat SARS-CoV SCH014. They also recorded the first live bat SARS-CoV, which they named WIV1, that they got from bat SARS-CoV RS3367, which showed even closer morphology to human SARS, which proved that bats were indeed the natural reservoir, and specifically horseshoe bats, and that they were capable of causing direct infection with no intermediate host needed. Now, here's where things got even more interesting. In 2014, Shi Zhengli, a virologist of the Wuhan laboratory in China, came together with Ralph S. Baric, the creator of Remdesivir, and created 2 new chimeric viruses from the bat SARS-CoVs that she discovered in 2013 in Hunan in Chinese horseshoe bats. Now, they claimed that they were creating these viruses to see if the bat SARS-CoVs could efficiently replicate in human cells, which confused me because I thought that the discovery in 2013 of these bats and the finding that they could use ACE2 as a receptor binding proved that they could infect human cells. So what was the purpose of coming together to create chimeric viruses to further prove that they could indeed do so? That I don't fully understand. Now, also, what happened in 2014 was a funding pause on any research going on for SARS, MERS, or avian influenza viruses, because some scientists thought that this could be risky and could cause a potential pandemic if any of these viruses were to escape or leak from the lab. So there was a lot of debate going on whether this type of research should be able to continue, which of course Barrick was in heavy disagreement about with the discovery of these two new bat SARS-like coves, because they had the potential for emergence as pandemics. So stopping research on SARS, MERS, avian influenza would stop any type of research on finding antibodies or vaccines that could potentially help stop a pandemic if one was to emerge from something like these bats. So they were allowed to continue their research because they had started before the pause happened. Remdesivir was initially being used for experimental treatments of Ebola. It was put in the running with 4 other drugs, including ZMapp. But ZMapp and Remdesivir was taken out of the running for Ebola because Remdesivir and ZMapp killed more patients than the other 2 drugs in the running. Now, in the same time that Remdesivir was being patented for Ebola, it was also being patented for coronaviruses, because Ralph S. Barrick had tested some of these antibodies on the chimera viruses he created from those bat SARS-like coves they found in 2013, and found that a combination of these drugs did help neutralize the virus. So he determined that a combination of these antibodies would possibly work against any pandemic emerging disease like COVID-19. But I had a theory, and unfortunately this theory came from watching the movie Outbreak. If this virus was created in the lab and you did test your antibody on it and it did work, once that virus was entered into human cells, it then would probably mutate. So whatever antibodies that were supposedly working for the virus you created would probably no longer work once that virus was able to infect the human host and then mutate into a different strain of virus that those antibodies would no longer work on. And like we've recently just discovered, Remdesivir is not working for COVID-19. The other interesting thing that is making people believe that COVID-19 could have been lab made is the fact that the bat that they named as being responsible for COVID-19, bat RATG13, was found in 2013, along with the 2 other bat SARS-like coronaviruses that were discovered that were able to bind to human ACE2, bat SARS-like CoV RS3367 and bat SARS-like CoV SCH014. And since COVID-19 also uses ACE2 as a receptor binding, one would think that they would have tested this bat that they're claiming as being responsible for COVID-19 and been able to find that this bat SARS-like CoV also was able to bind to human ACE2. So why wasn't it reported along with these other 2 bat SARS-like CoVs in 2013? Instead, it was just left sitting in a freezer for 7 years. Horseshoe bats have now been implicated for both SARS 2003 and SARS-CoV-2, aka COVID-19. Now, horseshoe bats used to be sold in the wet markets where SARS 2003 outbreak occurred, but by 2019 they were no longer being sold in wet markets. Nor do horseshoe bats reside in the wild near Wuhan, nor do they hibernate in cities near Wuhan. But they are used for biological research, especially in labs in Wuhan. Now, on top of that, there has been widespread doubt on whether the virus actually came from the Wuhan market or was brought into the Wuhan market from a scientist who may have been infected in the lab and then had brought the virus with him into the Wuhan market, which then caused the widespread infection. Now, this comes from a report from a study that was done on 99 patients, where 49% of those patients had not had any contact with the Wuhan wet market. So this is what caused more suspicion on whether or not COVID-19 had escaped from a lab. Now, of course, this is possible to have occurred as we have already had a few times where infectious diseases have escaped from laboratory containment, like when the CDC exposed 84 scientists to anthrax, or the 1977 H1N1 human influenza pandemic, or smallpox outbreaks in Great Britain from 1963 to 1978, or the 1995 Venezuelan queen encephalitis outbreak in 1995, or the various SARS outbreaks that have occurred since after the SARS outbreaks of 2003 and 2004. Now, here's the thing that I found interesting throughout my research. I went back and I looked up all of the bats that were named to be responsible for such emerging infectious diseases like Marburg, polio, rabies, Hendra, Hanta, Nipah, etc. And most of these viruses were caused by flying foxes or majorly fruit bats. Now, there were 2 horseshoe bats named that caused diseases like Marburg and Hanta. But outside of that, no coronaviruses were ever found within bats until after SARS 2003. And you would think if bats are the carrier of such deadly diseases like Marburg and Ebola, that at some point in time a coronavirus would have been discovered within a group of bats. So why did it take so long before bats were discovered harboring these diseases? And the other thing is that in the 1960s, when they were trying to discover what the human common cold was, they found out that the human common cold had the same type of crown-like shape like the IBV in chickens. So the human common cold was giving the IBV-like virus name until they deemed it coronavirus. And they had determined that it had come from maybe a combination of bird coronaviruses and mouse coronaviruses. So exactly when did bats become harborers of coronaviruses? Now, Event 201, a global pandemic exercise that was run by Bill and Melinda Gates 1 month before the SARS-CoV-2 outbreak occurred in Wuhan, is really what got people thinking that this may have been a planned pandemic, as such things have been going on throughout our history for quite some time. But that'll be a topic for episode 3.